Today, I will be talking about the biochemistry behind cancer. By cancer, I mean the disease cancer and not the zodiac sign. There are so many things going on in a cancer cell, so I will only be focusing on the biochemistry alteration behind the Warburg effect and touch a little bit about the pentose phosphate pathway. First, we need to know what the Warburg effect is. Normally, when free glucose enters the cytoplasm of a cell, glucose gets converted to pyruvate. Pyruvate then enters the mitochondria to go through the PDH complex, and with the presence of oxygen, pyruvate becomes acetyl-CoA, which then enters the citric acid cycle. The products of the citric acid cycle includes 3-NADH and 1-FADH2. These products enter the electron transport chain to produce about 36 moles of ATP. Unfortunately, the PDH complex, citric acid cycle, and electron transport chain are not shown in this figure. In the PDH complex, if oxygen is not present, pyruvate is typically converted to lactate, which yields only about 2 moles of ATP. Now, in Warburg effect, pyruvate that entered the PDH complex still becomes a lactate even with the presence of oxygen. This is also known as aerobic glycolysis. As the figure shows, dividing cells or proliferative tissue can also undergo aerobic glycolysis, and not just the cancer cells or tumors. However, for the sake of this video, I will be focusing only on cancer cells. Warburg came from Otto Warburg. He was the first biochemist who first noted that aerobic glycolysis is a characteristic of cancer cells in the 1920s. He won a Nobel Prize for the discovery of cancer. Normally, the first step of glycolysis is the phosphorylation of glucose to glucose 6-phosphate. The transfer of the phosphoryl group from ATP to the hydroxyl group on carbon 6 of glucose is catalyzed by the enzyme hexokinase 1. In aerobic glycolysis, phosphorylation of glucose to glucose 6-phosphate still happens, but the enzyme that catalyzes the reaction is an isozyme of hexokinase known as hexokinase 2. Isozymes are 2 or 3 different molecular structures of an enzyme, but still with the same function. Tumor cells express hexokinase 2 enzyme. By the way, in this figure, I crossed out the pathway that we have not talked about in class. As we can see in the figure, hexokinase 2 enzyme is bounded to the mitochondria. Here, the enzyme has already access to any ATP generated by oxidative phosphorylation. Since it's already binding with the mitochondria, it is not susceptible to feedback inhibition by its product, glucose 6-phosphate. Feedback inhibition means that if there's too much glucose 6-phosphate, then the conversion of glucose to glucose 6-phosphate should stop. Hexokinase 2 enzyme is not susceptible to this kind of inhibition. Another biochemical alteration is that tumor cells express an embryonic isozyme of pyruvate kinase. The figure says PKM2, but for simplicity, we will just call it pyruvate kinase. In normal cells, the enzyme pyruvate kinase that catalyzes the reaction of phosphoenol pyruvate to pyruvate is a tetramer. However, in cancer cells, the pyruvate kinase is a dimer. This dimeric pyruvate kinase has low catalytic activity because it has reduced affinity for phosphoenol pyruvate in comparison to the tetrameric structure. Since the conversion of PEP to pyruvate is reduced, there will be a buildup of phosphoenol pyruvate or PEP. So you are probably wondering, if there is less pyruvate available, how could a tumor still grow? Or how could it still produce lactate? I will state the answer later, but here is a hint. The answer is in the question itself. If there is reduced conversion of PEP to pyruvate, there will be a buildup of PEP and other glycolytic intermediates such as glucose 6-phosphate, fructose 6-phosphate, GAP, and DAP, and 3-phosphoglycerate. We haven't learned this in this class yet, but the glucose 6-phosphate from glycolysis can be processed to yield NADPH in the pentose phosphate pathway. NADPH plays a very important role in cancer cell growth. So, what is the pentose phosphate pathway? It is a metabolic pathway parallel to glycolysis. It generates NADPH and pentoses, or 5-carbon sugar, as well as ribose 5-phosphate. I'm not going to dive deep in the pentose phosphate pathway since we have not learned this in class. However, the main takeaway is that cancer cells require ribose 5-phosphate for nucleic acid synthesis and NADPH for biosynthesis of macromolecules. NADPH has a different function from NADH. NADH carries electron to the electron transport chain, while NADPH is used for anabolic reactions or the synthesis of large molecules such as fatty acids and steroids. Tumor cells like to proliferate, and they could do so with the help of the pentose phosphate pathway. As mentioned in slide 7, buildup of phosphoenol pyruvate or PEP can also lead to the buildup of glucose 6-phosphate, and this is the precursor of the pentose phosphate pathway. The accumulated glycolytic intermediates are used by the pentose phosphate pathway to synthesize ribose 5-phosphate. Even with less pyruvate produced because of the dimeric structure of the pyruvate kinase, the tumor can still grow because the NADPH and ribose 5-phosphate produced during the pentose phosphate pathway helps in production of macromolecules and nucleic acid synthesis of the cancer cell. Also, lactate can still be produced, but in little amounts. Here are the benefits or reasons why cancer cells still perform aerobic glycolysis even if it only yields few ATP molecules. You can pause the video and watch it, but I don't have enough time, so I'm gonna move through it. Before I end the presentation, I just want to share what I found on YouTube on how cancer cells and normal cells proliferate. Thank you for listening.